Okay, so this set of lecture slides will be an introduction to cell signaling or signal transduction. Okay, so this is chapter 16 in the textbook. So we want to keep in mind that just like with your cell phone where you get a radio signal in and then a sound out, cells work in the same kind of way. So they're going to get an extracellular signal from a molecule. It's going to bind to a receptor, and then that gets translated into an intracellular signal and then an output where we're going to get some kind of effect response by the cell. So you can see in the figure above, these are yeast cells, and you can see that phenotypically when we look at them, they look very different. So in panel A, they have a nice round appearance, but once they get the signal to actually start elongating and budding off because of growth factors that are present, their appearance changes, and this is because there's all this response that occurs, rearrangement of cytoskeleton. So some of this information links with our other chapter that is in this unit 3. So our other chapter will be chapter 17 that looks at cytoskeleton. Okay, so again, this chapter looks at cell signaling, or also known as signal transduction. And again, it's that input in, information's being taken in, processed, and then an output. So cells can communicate in various ways through those signaling molecules. Okay, and how far that signal has to travel also affects how the method, the way in which the cell is communicating. If you've taken anatomy and physiology, you would have talked about some of these. We've already talked about neurotransmitters, so we've already kind of introduced the concept of cell signaling when we were talking about the generation of the action potential and then spreading that across to another cell through that synapse. Okay, so looking at this figure, in the first panel, panel A, we have the endocrine, right? So if, again, taking A&P, you would have talked about the endocrine system, and this is where we have hormones. So hormones are going to be produced by an endocrine cell. They get secreted out, and then they travel through the bloodstream to then act on a target cell. Okay, and so that target cell has to have a receptor for that hormone in order for then the signaling to occur with inside that target cell to then cause a response. Sometimes that response is by producing and secreting another hormone or producing an effect like proliferation of cells. Okay, so we have growth occurring. Another way in which cells communicate is paracrine. So paracrine is where the target, the signaling cell is going to produce the molecule, the signaling molecule, and it's only going to travel a short distance, so it's neighboring, nearby cells, close cells that have the receptors that will be the target that then you would see the effect occur. With a neural communication, this is where we're going to have neurons acting on a target cell. So this could be a neuron to neuron, or it could be a neuron to like a smooth muscle or skeletal muscle where we would get like a contraction. But we have that synapse that neurotransmitters are going to go across. So again, stuff we had talked about in Unit 2, so keep that in mind. Okay. And then we can also have communication that requires contact dependent. So this is where the signaling cell is going to have the signaling molecule bound to its membrane, and then it would interact with the target cell that has the receptor. So it's not a release of the signaling molecule because that signaling molecule, again, stays membrane bound to the signaling cell. So they have to be very close together, right? So if we think about from a distance standpoint, contact dependent those cells are going to have to be very close together for those interactions to occur. With neural and paracrine, they can be a little bit further apart, So there doesn't have to be that kind of direct contact. And then endocrine, these can be very far apart. So, I mean, if we think about the endocrine system, we have hormones that are produced in our brain that then affect our pancreas, right? So pretty far distance. This table from your textbook just shows some of the different signaling molecules. So again, they fall into several different categories. Hormones would be for the endocrine, local mediators, so those would be for our paracrine, neurotransmitters for our neural, and then we also have contact-dependent signaling molecules like delta. Okay, and you can, I would highly recommend looking at this table, knowing an example from each one. I'll be honest, I kind of think of insulin for hormones just because of my research in diabetes. Your different growth factors, so depending upon what cell it's targeting, that usually would indicate the growth factor it is. So epidermal growth factor is going to act on and stimulate the epidermis. Platelet-derived growth factors are the source of origin actually is from platelets, and they'll act on different cell types. So just like in science, everything doesn't fall the same way. If you want to think about from the last unit, we talked about mast cells and how they have exocytosis. So histamine would be a great local mediator. So you have these mast cells that release the histamine, and they're going to have effects close by. This is why you get hives, right? And then we talked about acetylcholine as a neurotransmitter. So again, just I would definitely, I'm not saying memorize the whole entire table, just get familiar with some of these different types of signaling molecules and what are their actions, what are their effects. And so you can see that those actions can vary, but in many cases it's we think of this as a stimulating, but keep in mind that we can actually inhibit. So if we look at under local mediators, transforming growth factor beta, TGF beta, it actually inhibits cell proliferation. So it's not always an excitatory response. It's not always like an activation of proliferation. Sometimes it's a feedback where it's a halting, okay, inhibiting pathways, inhibiting proliferation, okay. So when we look at cell signaling, we need to have receptors, and so receptors can either be found on the cell surface or they can be found intracellular receptors. And so if we look at hormones, again as an example, there are hormones that bind to cell surface receptors because they can't get into the cell by themselves, or they don't need to because the receptor they bind have signaling molecules that allow for signaling a response to occur inside the cell without them actually entering. So if you have a hydrophilic signaling molecule, those tend to have cell surface receptors, where if you have a hydrophobic signaling molecule, so some hormones are hydrophobic, so they actually will go inside the cell, and in that case their receptors are intracellular. Okay, and in many cases that receptor and the molecule actually get transported into the nucleus, and then they cause gene expression changes. So again, if you've taken A&P, when you talked about steroids and how they work, there were steroids that would bind to surface receptors, and then there are hormones that are going to bind intracellular receptors. So just to give you an example, insulin receptor would be found on the cell surface, so it's going to bind insulin and then bring that in through signal transduction, cause and effect. Okay, where with an example of an intracellular receptor would be like estrogen receptor, so that would be found inside the cell. So we also want to keep in mind that signaling molecules don't have the same response for every target cell. So a signaling molecule like acetylcholine can bind to different types of cells. So as we can see in the figure, here's 3 different types of cells it binds to. But each one's going to have a different effect, right? So if acetylcholine binds to a heart muscle cell, it's actually going to decrease the rate and force of contraction. So it's going to augment how the heart is beating. Acetylcholine, if it binds to a salivary gland, it's going to cause the secretion of saliva and enzymes. And if it binds to skeletal muscle, it's going to bind to those ion channels so that they open, allowing for flux of ions like sodium. Okay, so that we would get contraction. So again, each target cell can have a different response. Also, it's not that the cell is just taking in information from one signaling molecule. So a cell depends upon multiple signals in order to determine what the response is. So it's like taking, a teacher is going to listen to every single one of the students, all the different questions, and then make a decision on what's best for the class as a whole, right? So here we have, in this figure, we have a cell that's taking in information from single molecule A, B, and C, and then it ends up having gene expression that relates to survival. Where if it receives the same signals plus D&E, then it actually says, okay, we'll grow and divide. Okay, but if we change, we have those same A, B, C, but now we have E and F, it actually tells it to differentiate and become a different cell type. And if there's no signal, that can signal the cell to die. Okay, different signals can induce apoptosis and catalyze to die too. So again, it's a collection of that information. So just like we saw with neurons can take information from different neurotransmitters and then come up with a response based off of what the combination is that they get. And if you're taking other classes, if you're taking in the future taking other classes such as like immunology, how a cell decides to respond is very much like this. It's taking in all the information. So for immune cells, they're looking at all the cytokines, and it's not just necessarily like X amount of this cytokine is going to cause it to respond this way, because there could be cytokines that will actually inhibit that response, not directly but indirectly, because again, the cell's kind of like a computer taking in all this information and deciding how the end response should actually be. And that response could either be slow or fast. So fast responses can be, less than a second, a minute. Other responses are going to be slower, and it's going to take minutes to hours. The other thing is with some responses, some signaling molecules are, kind of over and done quickly, where other ones can have a more prolonged effect, so that you have a more sustained response. And that's really important with the hormones, because there's certain homeostasis states that you want to you don't want to have to constantly produce hormones. You want those responses to be maintained, so you have homeostasis, so that other responses are only during like times of stress. So you have your stress responses, which are going to be short-lived, because the idea is that stress event is not prolonged, so that release of stress hormones tend to be more short-lived, because again, eventually, the woolly mammoth is going to stop chasing you, and you can then focus on digesting. Okay. But again, if we think about these responses, if we have a signal molecule binding to the surface receptor, we then have the intracellular signaling, as we can see here, and there are arrows, and we can have different types of response. We can alter protein function. So maybe, you guys saw in your proteins that some of them were in the cytosol, but sometimes it's on the cell surface, and so maybe that changes where it's being expressed. So maybe, if you have a KDL, that KDL now gets removed so that you can go to the surface. It can also alter protein synthesis, so we can have an effect on RNA production or the stability of the RNA that's being produced. So we can either increase protein expression of genes, or we can decrease protein expression of genes. And then we can also have altered cytoplasmic machinery, which means we can have more changes in vesicle docking, rearrangement of cytoskeleton, and just this kind of altered cell behavior. So if we think back to that first slide, not the very first slide, the second slide, where we had those yeast, right? Like there's obviously cytoplasmic machinery being altered, and looking at the cytoskeleton and rearrangement of the cytoskeleton, because the response lends to that, right? And so, and some of that then lends to altered cell behavior, right? So again, if you think about immune cells, how they behave after they've been, that signal's been triggered, you can go from an unactivated T cell to an activated T cell, and that's going to change how it behaves in its environment. Everything to attacking and trying to kill a pathogen, to assisting and helping other immune cells. So if we look at this overview of cell signaling, this figure kind of lays out a general basic scheme of cell signaling. So this is a good figure. I would hope that everybody in this class will have a strong understanding of this figure. And part of your PET cell assignment will be looking at how your protein, your PET protein, fits into this for your, now we're moving to the PET cell, okay? So you'll have this firsthand experience on how this cell signaling works. But again, we have to keep in mind that there's going to be a single molecule, extracellular. It's going to bind to receptor protein. That receptor then, we'll talk about some different signaling pathways, then has intracellular signal molecules that usually have like a regulation in them. So it's not just one molecule has to get activated and then we have a response. It's a cascade, so we can make a decision. The cell can make a decision about whether we carry forward or not. Okay, those intracellular signaling molecules then will affect effector proteins. So this could be some kind of enzyme or a cytoskeleton protein, or maybe a transcription regulator, right? And so depending upon what effector proteins are affected, we're going to have a different cell response. And so this is, if it's a metabolic enzyme that's being affected, then we'd have altered metabolism. If we affect a cytoskeleton protein, then we're going to alter the cell shape or movement. And if we have transcription regulator, this is going to alter gene expression, right? And so again, if we look at any cell signaling pathway, we would be able to fill in this information for that. Okay, so we'll end up talking specifically about 2 cell signaling pathways because that's what your textbook points out. There are additional ones, and we may end up looking at them. I would really hope we would look at 1 or 2 of them, but it's kind of like I hope everybody understands G protein coupled receptors and receptor tyrosine kinases. But then we'll probably do like a little special interest class assignment next week where we'll look at some of these other ones in small groups and kind of report back about them. Okay. But again, any of these pathways, we could kind of superimpose those pathways onto this overview and see how those specific proteins that are involved with each of the pathways fall. Okay. So again, this is a really nice general slide, so that if, as we're talking about the different pathways, if it's overwhelming, if it's too much, come back to this one and just, think about it, simplify it. Think about, okay, well, where do the G protein coupled receptors fall? Where does the receptor tyrosine kinase fall? Where did my molecule fall in this pathway? I know it's involved in the Wnt pathway, but where does it fall in the Wnt pathway? Okay. So this is a list of some of the common signaling pathways. Again, as I just mentioned, we'll be focusing on the first two, the G protein coupled receptors and receptor tyrosine kinase. Those pathways, because your textbook picks those out. I know some of you have your PET protein falls in those categories. Some of you have PET proteins that are in different signaling pathways, so don't worry, you'll get to look at them. And again, as I mentioned, we'll probably do a class group assignment where you'll look at some of these other pathways, so that you'll have an active repertoire of at least 2 to 3 different pathways. And again, it's going back to that last figure, and how can we generalize these pathways so that it's not so overwhelming. Okay, so I guess I should have said in the beginning, cell signaling can be overwhelming. Most schools have a course that's just on, it's either called cell signaling or it's called cell communication. But when we look at cell communication, it's really, in some ways, it's looking at an extension of cell signaling, right? So how cells are communicating with one another. Okay. In this lecture, I just want to provide a brief overview of these two pathways that we're going to look at. There are going to be separate audio lectures on those pathways. Will be in more detail. The reason I'm doing that is because if you find those other lectures overwhelming, you can come back to this lecture and you can say, okay, well, at least I know this about G protein coupled receptors, and then go back to the other lecture and say, okay, I want to add this information into my understanding. Okay. So with G protein coupled receptors, sometimes abbreviated. Okay, so for the G protein coupled receptors, it's one of the larger families of signaling molecules. So you can see here in this figure, we have our G protein coupled receptor, or GPCR. And so one of the identifying characteristics of it is that it has these 7 transmembrane domains. So it actually is known as a 7-pass transmembrane protein. And so that's the common feature, but they can be very diverse on what ligands they can bind. So that the receptor portion of that, where it recognizes the signaling molecule, can vary between the different GPCRs. Okay, and so as far as what can act as signaling molecules, it can be things from odors, pheromones, neurotransmitters, small peptides to large proteins. And so because different variants of the receptors are involved in disease, they're commonly looked at targets for drug therapy. So can you design a signaling molecule that will bind the receptor to block its effect? Because then you would block downstream response by the cell. Okay, and again, we'll go into more detail and talk about this when you listen to this audio lecture specifically about G protein coupled receptors. So they're going to get their name because there'll actually be a G protein that it is going to bind. And again, we'll look more at that in that specific lecture. So just some, again, the interesting characteristics of a G protein coupled receptor is that it's going to have the 7 transmembrane, and then it's going to have these intracellular or cytosolic or inside loops to them. So there's going to be 3 loops, and then it has a tail. Okay, so it has a C-terminus tail. It also has an N-terminus tail that's going to be extracellular. So it's also going to have 3 extracellular loops also. Okay, so again, these are kind of some of the conserved features of the G protein coupled receptors. And we'll talk more about these later on. I guess another thing I'd want to point out is that there's a lot of post-translational modifications that happen on the receptor. And so one of the reasons I include this figure is because this figure labels different post-translational modifications. So you can see that there. There's different sites of phosphorylation. So those sites of phosphorylation affect the ability for the receptor to activate and to bind to the G protein. You also notice that there's a lipid raft localization signaling sequence, so that allows it to be found in those lipid rafts, and so you can see how there's that cholesterol. So this would be an area where we would have a lipid raft. So they're going to be found kind of clustered together. And we had mentioned in the first unit how lipid rafts are important because these are going to become these islands on the cell membrane, or on the plasma membrane, where we'll get these signal transductions, cell signaling pathways occurring. Okay, so if we want that to happen in lipid rafts, we need to make sure that these proteins, these receptors, get to those areas of the lipid rafts. So the other pathway we're going to look at is receptor tyrosine kinase, RTKs, and so these are another large family of proteins that are going to bind a diverse group of cell signaling molecules, just like with our G protein coupled receptors. These are going to be also involved in diseases, so different mutations that affect binding can lead to different disease states, and so they're also researched quite a bit, looking at drugs that target them. You'll notice that they look different because they only have a single transmembrane domain in them, so they're known as a single pass transmembrane protein, opposed to the 7 that we see with the G protein coupled receptors. Okay, this receptor is important early in embryonic development and also for cell survival. So obviously, if it's affected, it can lead to apoptosis. If it's affected, it can lead to different failures in embryonic development. Okay, so again, those are the 2 pathways we're going to look at. So at least now you have an idea of what the receptors look like, and we will will be 2 additional lectures for you to listen to that are specific about those pathways. But I would say definitely make sure that you feel comfortable in this introductory information. And if you have any questions about it, please send me emails or ask for me to cover or go over the information in class. Okay? Thank you.